Good morning, good afternoon, and good evening. Welcome back to Chapter 16. We're doing the survey of life. Remember, the survey of life is we're going to start with the simplest organisms and talk about them each in turn until we make it up to the most complicated organisms. We're talking about prokaryotes at the moment. We're talking about bacteria and archaea. We just got done talking and saying that the biomass of prokaryotes is 10 times the biomass of all other living things combined. There are lots of bacteria. We refer to it and say that we live in a bacterial planet. Everywhere you go, there are bacteria. You can't get away from them. Most of them are harmless. We said that. Only about 1 in 1,000 species of bacteria are harmful. Most of them are benign, which means they're not good or bad. And a few species are downright beneficial. One of the groups of bacteria that are beneficial are decomposers. One of the jobs that prokaryotes accomplish is by recycling waste, by breaking down the dead bodies of plants, of fungi, of animals, and turning those nutrients, or rather returning those nutrients, returning those nutrients back to the biological system, back to nature. These recyclers can also not just recycle biological waste, poop, feces, dead organisms. They can also be used to recycle chemical elements, chemical wastes. There is a saying that for any chemical that there is out there, there's a bacteria that will eat it. There's a lot of promise in cleaning up chemical wastes using bacteria. One species of bacteria lives symbiotically with the roots of legumes. Legumes are bean plants, soybeans, green beans, peas, peanuts. There's one species that live in the root nodules of these plants. The root nodules, if you pull up these plants, they've got knots. They look like tumors on the roots. And living inside of those tumors are bacteria. And the bacteria are not harmful, they're actually beneficial, because what they do is they pull nitrogen out of the atmosphere. Most of what you're breathing in right now is not oxygen. What you're breathing in right now is air. Air is a mixture of many gases. The primary gas that you are breathing in is nitrogen. About 71% of air is nitrogen. What do we do? What does our body do with gaseous nitrogen? Nothing. We can't do anything with it. There's only one species of organisms, only one species of life on the planet that can do anything with atmospheric nitrogen, and it's these bacteria that live in the root nodules on legumes. They're able to take that atmospheric nitrogen and convert it into a usable form. They can convert it into ammonia. Once they convert it into ammonia, it gets converted into other products, and basically it becomes a fertilizer for the plants. If you've ever had FFA, if you ever learned anything about agriculture, you know that you have to rotate your crops. And about every second or third planting, you should plant a legume. Around here, it's usually peanuts. In other places, it's soybeans. You should plant a legume because it will add nitrogen back to the soil. It will reduce dependence on nitrogen-heavy fertilizers. Crop rotation. Why do you want to plant a bean of some kind? Because it adds fertilizer to the soil. As we already said, there are two domains of prokaryotes, eubacteria and archaea. Within domain eubacteria, remember we talked about this in the first video, eu means what? It means true or good. So eubacteria means true bacteria. Eubacteria come in three shapes. The three shapes are cocci. Cocci means berry. What is a berry like? What is a blueberry like? It's a sphere. So cocci means sphere, spherical. The next shape is called bacillus. A bacillus means rod, actually means little rod. So a third shape of bacteria is a rod shape. Kind of like a sausage shape, actually, give or take. And then the last shape is spiral, like a corkscrew. And there's two kinds of spirals. There's spirilla, which is a short, rigid spiral, and there's spirochetes, which are longer and more flexible. Here we see different shapes of bacteria. This is that cocci. Notice that definite berry shape that we see. This is bacillus. Well, I guess, you know, that looks more like a Twinkie than it does like a sausage, right? I've been accused of my analogies being often food-based. Look, man, I just try to pick stuff we can all identify with, and I think we can all identify with Twinkies. And last is spirochete. Spirochete is a spiral. This is the long, flexible. Spirilla is much smaller but shorter and less flexible. One of the characteristics of domain eubacteria is that they all have a cell wall that is made of a very specific substance, and this very specific substance is called peptidoglycan. If you had me for Biology 1, you know that one of the things that's important to me is breaking down words to make them understandable. We've already done it today by breaking apart eubacteria. Well, let's break apart peptidoglycan. You're in Biology 2, which means you passed Biology 1, so some of these words should be familiar. For example, glyc. Glyc means what? Glyc means sugar. Peptide, we talked about what that had that name in it, that had that word in it in Biology 1. We talked about peptide bonds. You may not remember exactly what that was, but a peptide bond is a bond between amino acids. What do you make by linking together amino acids? You make proteins by linking together amino acids. So peptido, it doesn't directly mean protein, but it certainly is related to protein. So if we break apart this word called peptidoglycan, we get two words, protein and sugar. Well, which is it? Is it a protein or is it a sugar? And the answer is it's both. Better question. If it's both, which is the primary structure and what's the thing attached to it? Always, when we see big, complex words like this, the thing that it is the last part of the word. So this is a sugar. What is the thing that is attached to my sugar? The thing attached to my sugar is a protein. So it's a sugar with a protein stuck to it. It's a very special sugar with a protein stuck to it, and it's called peptidoglycan. The only place we find peptidoglycan is in the cell walls of bacteria. It's the only place you find it. That's one of the requirements to being a bacteria, to being within domain eubacteria. If you're in domain eubacteria, it's because you have a cell wall made of peptidoglycan. The job of this cell wall is pretty much the job of any cell wall, and that is to protect the cell, to be a physical protection against invaders. And it can also prevent the cell from bursting. Bacteria have to live in water. Remember, when a cell is in a hypotonic solution, water will rush into that cell. And if it doesn't have some sort of barrier around it, if it doesn't have a mechanism for regulating the water coming in, it can actually cause the cell to rupture. It can cause the cell to burst. Well, if I take a water balloon and overfill it, if I keep putting water in it, it's going to pop. But what if I take that water balloon and I put it inside of a wooden box? And then I fill it up. Will I pop the water balloon? And the answer is no, the water balloon won't pop. And the reason why it won't pop is because when it reaches the point to where it should burst, it's already touched the sides of the wooden box, which prevents it from getting any larger. So it prevents it from filling anymore. That's one of the jobs of this cell wall. The jobs of the cell wall is to prevent the cell from popping, from bursting. Here we see a diagram of a bacterial cell. Again, I hope you've got the PowerPoint downloaded so that you can look at this up close. It might be difficult to see detail in the video. We've got a number of structures that are around the outside of my bacterial cell. My outermost layer is called the capsule, or slime layer, and that's this thing with these little sticky things coming off of it, these little fingers. They're called pili. This capsule is a sticky, sugary layer on the outside. It depends on the species as to what its job is and what it does. The capsule. Underneath of the capsule, we've got the cell wall. So I'm not sure if you can see it very well, but this red layer that goes around right there, that's the cell wall. That's that stuff made of peptidoglycan. If we go in one more layer, we get to that plasma membrane, that cell membrane. Remember in Biology 1, we talked about how all living things are made of cells. And all cells have a cell membrane. All cells have a plasma membrane. So bacteria are cells, so they have a plasma membrane. So here's that cell membrane right there. And then once we get inside, now we've got the internal guts of the cell. We have its chromosome here in the center. We've got those ribosomes we were talking about before. And then we've got a special little thing called a plasmid. Now, not all bacteria will have plasmids. Some do. What is a plasmid? A plasmid is a miniature bacterial chromosome. If I were to take this big clump right here and unwind it, it would be a big circle, a big loop. We see the plasmid is just a small circle, a small loop. All right, so domain eubacteria. The capsule is that outer layer. Its job is to stick to other things, to stick bacterial cells together and to stick the bacterial cell to other things. And it also can shield the pathogen from attacks. Lots of things want to eat bacteria, and it's difficult to get a hold of it because of this sugary, slippery layer.